In this video, we'll talk about cell lines and cell strain. Cell line is the general term that applies to different populations of cell that can be maintained in culture in vitro. This is a portion of the intestine, so we can culture these intestinal cells as a cell line, and we can call it an intestinal cell line. There are different features of the cell line, such as they can be maintained for an extended period of time. It should be retaining stability in terms of certain phenotypes and function. Cell line is generally clonal. That means from the parental cell line, there could be clonal progeny. When it comes to feature of the cell lines are generated from subculturing a primary culture. And the cell line usually display functional similarities with the primary cell with certain degree of dissimilarities in terms of genotype and phenotype. There could be modifications. Cell lines are divided into two groups based on the growth patterns. So finite cell line divides for limited number of times, whereas the continuous cell line divides for infinite numbers of time. Finite cell line has limited division capability. That means they would divide for 20 or let's say 100 times for a finite number, and they would not divide anymore, they would eventually die. And the time or the number of times they divide before dying is governed by several factors such as cell lineage differences, species, culture conditions, and media conditions. Now let me tell you that finite cell lines are generally adherent in nature. That means they would stick to the surface of the culture plate. Now, infinite cell line, in contrast, can grow for indefinite number of times. They generally divide faster compared to a finite cell line. Now, these cells can eventually grow on top of one another, and they are forming a multi-layered structure like a tumor. So, infinite cell lines are pretty much like a tumor or a cancerous cell. Now, generally, normal cells are obtained from tissue or organ, and you form a primary culture out of them. Primary culture is then subcultured. After subcultured, you generate cell line. From that cell line, you can select a portion, a subset of that cell line, and you can make them immortal and make them an immortalized cell line. Or also, you can take the cells from a cancer patient and make a cancer cell line which are immortal. So, moral of the story, cell strain is one subset of a cell line, and they are selected based on specific characteristics. Cell strain will often undergo additional genetic changes compared to the parent cell line. So, moral of the story or the take home message is the cell strain is one kind of subset of a cell line. The first continuous cell line ever established was the HeLa cell line, taken from Henrietta Lacks, who was a carcinoma patient, cervical carcinoma patient, and this was the first aneuploid line derived from human tissue. And so far it has been used for several purposes throughout the world. Now let me tell you the example of cell strain. HMC1 cell line is a frequently used cell line to study mast cell. So basically this is mast cell leukemia cell line. So whenever we want to study some aspects of mast cell, we can use this cell line. Now there are two variants of this cell line, HMC 1.1 and HMC 1.2. Both these cell variants are actually the cell strain or a subset that are selectively selected differently based on specific mutations. Okay, let's talk about the applications of cell line in research. In molecular biology or cell biology lab, cell cultures are kind of bread and butter technique. Every day people are doing cell culture. Now let's say you have figured out a particular novel protein, protein X let's say, and wanted to understand where this particular protein is localizing inside the cell. So you can clone that particular protein in an expression vector, transfect it into the cell line, and then ask the question where this GFP bound protein is localizing. And you can get your answer by doing some microscopy technique. In this case, you can see the protein is localized in the nucleus because the fluorescence is coming from the nucleus. This is one type of question. There are many other questions that can be answered in a research settings. For example, you have discovered a drug which might have an anti-cancerous property. How do you test that? You can use some cancerous cell line, and in one set you don't treat it with anything, and in another set you treat it with this particular drug, and then you check viability for both these cases. If you see a decrease in viability, that means your drug might be very efficient. People also create efficacy curves from this particular assays. There are many applications of cell line in industry. Let me tell you one of that. So whenever you need antibodies, you order from the companies, and the companies generate antibody using hybridoma technology, which is a way of generating monoclonal antibodies. In hybridoma technology, a hybrid of antigen specific B cell is fused with myeloma cells. Antigen specific B cells can generate all the monoclonal antibodies, but they are finite, they are mortal, so they has to be fused with myeloma cells to produce a hybridoma. This hybridoma can be used to generate monoclonal antibodies generation after generation, and that is how these industrial companies can supply you with antibody years after years. Now let's talk about some clinical applications of cell line. So in clinical diagnosis, several cell lines are used. One such example is to detect presence of anti-nuclear antibodies in patient's blood sample. For example, if a patient has systemic lupus erythematosus, they might have particular anti-nuclear antibody in their blood sample that can be detected using this HEP2 cell line. HEP2 cell line has all the native protein arrays and hundreds of antigen, which provides an ideal substrate for ANA detection. So patient sample might have these autoantibodies, might not have them. Using this particular cell line and a combination of immunofluorescence, we can understand whether the patient sample has anti-nuclear antibodies or not. Looking at the patterns, there could be several clinical interpretations that can be made from these immunofluorescence data. So that is why usage of cell line is crucial in medical diagnosis as well. And we gave just one example. But cell lines are very different from a native tissue. Let me tell you why. Let's say you have a portion of the brain, a neural tissue, that would have different cell types. They would have ABCD neurons, different type of glia, astrocytes, oligodendrocytes, etc. But a neural cell line would have isolated or clonal population of only neurons. That means they would create synapses, but the way they create synaptic strength, properties might be very different from a native or a primary tissue. That is why when using cell line, we have to think about some limitations. And this is why people these days generate 3D cultures which mimics more like organ. And these are known as organoids. For brain, cerebral organoids are generated. For many other tissues, organoids are generated. If you are interested, you can click on the I button to know more. There are several limitations of the cell line, as we have mentioned, and we should know this because when we are using cell line for our own usage, we have to understand the limitations. First of all, serial passage or subculturing leads to acquisition of several mutations over time that can lead to genetic drift, and that can cause several heterogeneity in the culture at a particular time point. So obviously, cell lines has many limitations. Cell lines never recapitulate the entire property of a primary culture. It recapitulates only certain aspects. Immortalized cell lines might have different chromosomal abnormalities, aberrations, and mutations. So they do not permit studying several biological processes, and we have to be very careful when we think and design our experiments using these cell lines. So you can get notes and flashcards regarding this topic and many other topics in my Facebook channel, in my Facebook page. So please share the link of my Facebook page with your friends. You could get many dynamic flashcards which would help with your preparation and understanding. As usual, don't forget to like, share, and subscribe. You can support my channel on Patreon, Super Like, or if you're an Indian viewer, you can support me via Bhim UPI. You can scan this QR code to pay me. Anyway, my courses are also present in Unacademy, which is India's biggest online learning platform. You can use my code AP10 to get a